It's a great honour to give this talk, and I'm going to be talking about exploring channels, slopes and bindings, pharmacological skiing, and I hope that's going to be obvious as I get through this talk. I've been at UCL for 21 years now, and at the moment I have some key collaborations with Shaheen Moldina at the Great Ormond Street and Professor David Abraham at the Royal Free. I'm proud to work for an institution that founded its principles on allowing education of students of any race and any religion, and was the first university in the UK to admit women on an equal footing as men. These are my disclosures, and I look forward to collaborating with AstraZeneca. Now, I'm not known for, I'm known for sort of rushing through my acknowledgements, so my staff said, please show this first. And I'd also like to acknowledge Brendan Whittle from the William Harvey Institute, and he's been a key collaborator and mentor for our laboratory, and he knows a lot about the history of prostaglandins. Now, I'm going to take you on a journey because I think this has really shaped the sort of person that I am today. So, the Clapp family came from Devon, and Roger Clapp took a trip on the John and Mary boat from Devon to Massachusetts. He came back to England to convince his cousin, Thomas Clapp, my 11th generational grandfather, to come to Massachusetts, presumably to set up a life without interference from the government. On their journey, they were shipwrecked, and the Clapp family actually survived the shipwreck and ended up building the town of Dorchester in Massachusetts. My grandfather, Professor Charles Clapp, was a geologist who studied at MIT. And later on, he got his PhD in 1910 from MIT, and then a few years later went to become the Vice Chancellor of Montana University. And he was a very able person. He didn't let the Depression allow to get into the way of building up Montana University, including building accommodation for the entire student body, because he felt that students should be part of an organisation that had help from the lecturers and tutors at the university. He also established the first freshman week, and many of you will know what that means to students as they come to university. Now, my father, Daniel Clapp, also studied at MIT. He was an organic chemist. And he studied C57 black mice and looked at the cancer-generating properties of the hydrocarbons. Now, C77 black mice, you'll know, are a strain of mice that are actually quite resistant to tumours. But he discovered that the hydrocarbons that produce cancer in these mice were probably producing it because of the benzene ring. So clearly, my love of chemistry is probably genetic. So my mother, Anne Hutton, she was a manager of consumer complaints, and here she is. Her pseudonym was Dorothy, and I don't know if the audience remember the TV advert from Mazola Oil, but she had to, and the TV advert said, Mazola Oil, it's like cooking your food in a safe, enhancing the food quality. And she actually had letters from people asking for the safe to cook their food in. Now, I have a love of sport from my uncle, who was an international cricketer. And as a family, my mother and uncle are related to Sir Leonard Hutton. He still holds the record for the highest number of runs in a test match, and I reckon the England team could do with him now. Anyway, so I think endurance is really a Clapp and Hutton trait. So I went to school at the Hinchleywood Comprehensive School, and I didn't actually dare to dream to go to university. Not many people went to universities from the school. I studied chemistry, maths, and physics, and I think I was the only other woman to study this combination. I wanted to study a subject that was translational, and I was very interested in pharmacology, as I thought that was much better than just studying straight chemistry. So I applied to Bristol University, and although I got the second highest grade in the school, I didn't quite make the grade, but Bristol University decided to take a chance on me, and I'm an ABBA fan. Anyway, I was a bit like a fish out of water, really, at university, because I didn't even have O-level biology or A-level, and I had to kind of, in 3 years, go from here to here and try and learn all the physiology I could muster. Anyway, I went and studied. I was allowed to have 3 years in a halls of residence called Churchill Hall. I was the first set of women that were allowed in, which was very helpful for my studying. Here I am, studying with books and also the dried milk and coffee. So my family would probably say, what's changed? Looks as well as my lifestyle. Anyway, because women were actually a rare species, and so I became quite popular with male students. And I was sent this Valentine's card, signed by 12 or 13 students, some of whom I still know. Others didn't really want to admit who they were. Love you too. Anyway. So I did actually graduate from Bristol University. And this is a picture of my mother and stepfather. And I went to work at St George's Hospital Medical School to do a PhD with Tom Bolton. Now, this is what Tom would tell the students that came into the lab. Well, you've got to, there really are no shortcuts in doing a PhD. You've got to understand, 100% of what you're doing. Be prepared to work for long hours. Well, I worked on a radioisotope that had a half-life of 12 hours. Therefore, I was working all night just to use the radioisotope. And stay focused. Don't let the administration get the better of you. Now, towards the end of my university degree, Virchow made this seminal discovery that the endothelial cells produced a relaxing factor. And it was shown by putting rabbit aortic rings in an organ bath and gently rubbing, and so that the response to acetylcholine would disappear. My job was a lot harder than Virchow's, because I was given the task of looking at endothelial-dependent relaxation from arteries that were very, very small, less than 0.5 millimeter in diameter. And I had to cut helical strips of these arteries mounted on micropins and have the endothelium intact. And that took me about 3 to 4 months to master. Anyway, I got nice relaxations to substance P and carbachol. And if I lightly rubbed the preparation, I would remove the response to substance P, but carbachol relaxation still remained. And this suggested that there may be more than one factor released from the endothelium. So Rick Lang taught me microelectrode recordings of blood vessels, and this took, again, about 3 to 4 months to perfect. And during this time, I had to become a decorator in order to fund the rest of my PhD. Anyway, to cut a long story short, I was really pleased to see that substance P didn't cause a membrane hyperpolarization, whereas carbachol did. And this suggested that there were, again, 2 factors released from the endothelium. And now we know that this is probably nitric oxide and endothelial-dependent hyperpolarization. Anyway, when I was working on this project, I was really interested to understand the ionic channels that were associated with endothelium-dependent hyperpolarization and how acetylcholine actually worked. And to do this, I would have to learn how to isolate cells and also to learn voltage clamping. And at the time, as a woman, I really wasn't encouraged to go along this path. And, I was really said, well, women just aren't biophysicists. Well, little did they know about our family genes. So this is a relative, a reasonably distant relative of mine, Cornelia Clapp. And she was the first professor of zoology at the Women's College, Mount Holyoke in Massachusetts. And she really brought research into this university. And people said, go and study with Dr. Clapp because she keeps frogs in a tank and lots of specimens. She also was the first scientist at Woods Hole. Many of you who have been to America know this is a premier research institute for marine biology. And she was an incredible woman because she got the first PhD in biology, but wasn't satisfied with just one PhD. She got 2 PhDs. And she worked on toadfish. And this is something that somebody said of her, that, she was a larger-than-life character, went in and out of the water, and yet this kind of behavior of a woman really wasn't that usual. So her motto was, there's no reason why you can't do anything. Anyway, I met Joshua Singer and John Walsh quite fortuitously at an international pharmacology meeting in the Barbican in 1984. This was just before I was finishing my PhD. And I really wanted to work with them because they were the first people to voltage clamp isolated smooth muscle cells. And so I persuaded them to go and let me work with them. So I made a similar journey to my ancestors, and instead of going by boat, I went on Pan Am Air to Massachusetts. I had no money, and I had to borrow money from my uncle. So what's changed? So I became a postdoc in the physiology department, and these two people were known as J squared, because the output of these two people were like the output of 4 academics. And they were also great mentors to me. And here is Josh Singer receiving an award some years later for mentoring. So the first task that I had to do was to build this electrophysiology setup. And they sent me to Worcester Polytechnic to learn electronics. And I did actually eventually begin to build a setup. And I think this kind of approach really helped me to become independent as a scientist. During there, I won a fellowship from the American Heart Association, and it was an incredibly productive time for me. So again, history is repeating itself. I worked on the toad stomach, isolated smooth muscle cells, and I worked with a guy, a Frenchman by the name of Michel Vivedou. He was a computer scientist and mathematician, and I was supposed to teach him biology, and he was supposed to teach me how to automate recordings of voltage clamp recordings in single cells. Anyway, so we used the patch clamp technique, and I think this is the first recording in smooth muscle showing 2 different types of calcium current, an L-type calcium current and also a transient T-type calcium current. And we showed that acetylcholine was able to activate selectively the L-type calcium current. And we also showed that the second messenger diacylglycerol mimicked the effects of acetylcholine. It wasn't all work. I used to take regular visits to the local ski mountain, Wachusett Mountain. And this is really pharmacological skiing by night. Now, the average temperature of skiing in Massachusetts was about -10 Fahrenheit, which was probably about -23 centigrade. So, and so clearly it made me into a hardy person. But really, I prefer skiing in the sun. We also made trips to Woods Hole, to the Society of General Physiology. And this is Steve Sims and Michel Vivedou, the public engagement of science on the beach. And this is a poster I presented there. Anyway, so when I finished my postdoc with John and Josh Singer, I went to work in Alison Gurney's lab at St Thomas's Hospital. Here is Alison winning an Athena Swan Award. And one of the crucial things that we managed to develop was a simple, efficient way of isolating blood vessels into single smooth muscle cells that were able to contract to noradrenaline, but also were calcium tolerant. And this was really key in order to try and look at the ionic currents in rabbit pulmonary artery. So this is probably one of my most cited papers showing that when you lower ATP from 3 millimolar to 0 millimolar in the patch pipette, cells became increasingly hypopolarized, and the effect of glibenclamide, which is a blocker of the K2P channel, became more marked in low ATP. And this just shows ATP levels are correlated with a more hypopolarized resting membrane potential. So Alison was an expert in flash photolysis, and so this is a method where you can put in an active substance into a cell and then use light to release the active substance. So in cells that didn't have any ATP in them, we put in caged ATP, and this is the sort of currents you got. You got lots of, you got background currents activated at quite high polarized potentials, and when you released ATP, the background current disappeared. And that really was one of the first key evidence for the existence of an ATP sensitive potassium channel in smooth muscle. And clearly at the time there was controversy as to whether this, the K2P was really a dephosphorylated form of the delayed rectifier. Anyway, over the next 14 years, I continued to work on the mechanism of how vascular K2P channels are activated, and I was awarded a Wellcome Trust Career Development Fellowship. And I moved to the Raine Institute in St Thomas's. Liking the Raine Institute so much, I went to the Raine Institute at UCL, where the head of clinical pharmacology was Patrick Vallance. And I really went there because he was looking at the role of INOS in septic shock patients. So a key collaborator was Mervyn Singer, a consultant of intensive care. And some other defining moments was a really good collaboration with Andy Tinker, who had an interest in the molecular assembly of potassium channels. So in the next couple of slides, we're going to try and summarize some of the key points or the key findings that I and others made. So I've shown you that lowering ATP activates the K2P channel and causes membrane hyperpolarization. I showed that these currents could be regulated or activated by nucleotide diphosphates. A postdoc in the lab was able to look at the molecular structure of the K2P channel in human pulmonary artery and showed that it was made up of the sulfonylurea receptor type 2B and KR6.1. And this really shows you a major difference between the K2P channel in vascular muscle and channels in the heart or the pancreas. So in an inside-out patch, levochromocalam, a K2P channel opener, doesn't work. But if you add the nucleotide diphosphate, you get channel openings. But a key thing that we discovered was that calcium, through the calcium-dependent phosphatase calcineurin, was a major regulator of the K2P channel. And that we showed that calcineurin inhibited the channel, but you could block this inhibition through using the immunosuppressant cyclosporin. Now, it was really interesting because Mervyn Singer came to you one day and said, I've got patients on cyclosporin and they have high potassium in their blood. And I kind of glibly said to him, well, I bet you that's due to overactivation of muscle K2P channels. Why not try glibenclamide? And this is what happens. So that he also had patients that were on nicorandil and isoflurane and got the same problem, life-threatening high potassium, or that will give rise to ventricular arrhythmias. So we tried glibenclamide, and it successfully treated these patients, and this was reported in The Lancet in 2005. Then a clinical fellow in the lab, as well as another PhD student, showed that induction of iNOS was a very good activator of the K2P channel, and that its activation led to vascular hyperactivity to catecholamines in endotoxic shock. What also led to some studies where in order to overcome the vascular hyperactivity to catecholamines, Alistair O'Brien and myself and Mervyn Singer suggested that terlipressin might be useful in the treatment of septic shock, and indeed it was. Now, what's the link between K2P activation and terlipressin? Well, vasopressin is a very well-known inhibitor of K2P channel function, so maybe that's how it's working in septic patients. And this is really some animal work showing that in control animals, norepinephrine is a potent raiser of blood pressure, and terlipressin really doesn't raise blood pressure very much. But after bacterial infection, your response to norepinephrine is much diminished, but you get this hypersensitivity to terlipressin. And another clinical fellow student in the lab showed that there was increased sensitivity of the V1A receptor. And that's kind of interesting because Ferrin, fairly recently, is looking at a selective V1A agonist, celipressin, in the treatment of septic shock. Okay, I'm going to slightly switch tack now and talk about prostacyclin. And, I managed to go skiing a couple of years ago and produced, in 10 days, a prostacyclin review with 200 references. And this really is my ideal lifestyle. So I was hugely jet-lagged during my trip. So I'd work from 3 to 8, go skiing, get rid of those aches and pains, and then go back to work. So let me talk about pulmonary arterial hypertension and the mechanistic basis of prostacyclin. For those of you who don't know much about pulmonary hypertension, what happens is that the blood pressure in the lung rises, in part due to a loss of prostacyclin and nitric oxide and elevation of vasoconstrictor agents like endothelin. So arteries become thickened, either due to smooth muscle growth or smooth muscle proliferation, and the intimal layer also becomes thickened. And then you get these plexiform lesions. So what we do is we take blood vessels from lungs and look at vascular reactivity. We also isolate cells from diseased parts of the lung, and this is just a picture of smooth muscle cells in culture. And then we compare the properties of these cells whenever we get normal human tissue or normal smooth muscle cells. And this work was really sort of started by a really good collaboration with Sheila Howarth from Great Ormond Street. She was a pediatric cardiologist. So prostacyclin was discovered in 1976, and soon after a synthetic prostacyclin was made, epoprostenol. And then some stable analogues that were based on the structure of prostacyclin. This is iloprost, and this is treprostinol. And then fairly recently, in 2007, another type of prostacyclin mimetic was made that doesn't have any of the structure of prostacyclin and is a prodrug known as Selexipag, and its active metabolite is MRE269. Prostacyclin is chemically and physiologically unstable, and it's rapidly hydrolyzed to metabolites that you can measure in the blood or the urine. And these prostacyclin analogues have variable stability in vivo, ranging from 20 to 30 minutes to several hours. Classically, it's thought that prostacyclin works by binding to the IP or prostacyclin receptor, elevating cyclic AMP. And what was shown by Lou Rubin in 1982 was that it was a potent dilator of the pulmonary circulation as well as systemic. And clearly by lowering pulmonary arterial pressure and lowering pulmonary vascular resistance, you will actually reduce afterload and therefore enhance cardiac output. Prostacyclin is also a potent antiplatelet drug. However, about 15 years ago, we were comparing the efficacy of different prostacyclins. And what you can see here is that the magnitude of cyclic AMP generation is highly variable and really not at all related to the binding affinity of these agents to the IP receptor, which is actually quite similar. And what we thought might be going on here is that maybe iloprost was known to activate EP1 and EP3 receptors. These are receptors that, so EP3 is negatively coupled to adenylate cyclase, and therefore might lower cyclic AMP, and EP1 is a receptor that elevates calcium and increases PD-1 activity. We also thought, well, maybe there's something else that treprostinil might activate that we didn't know about. And it was only really 10 years later that we began to shed some light on this. However, another observation that we made was that prostacyclins, under some conditions, can relax blood vessels through activation of potassium channels, and we really couldn't find evidence that cyclic AMP was the major cause of this relaxation. So here we have cicaprost-induced relaxation, and it's almost completely, entirely blocked by barium, suggesting that that cicaprost in this tissue, I think it's rat tail artery, was activating the inward rectifier. However, the response to forskolin was not at all affected. And if you blocked adenylate cyclase activation, we still couldn't block the effect of cyclic AMP. And we actually had a hard time publishing this because it at the time was quite heretical. But it's been known for a while that prostacyclins has other targets, these nuclear receptors. And this can be seen on the next slide. Hey, I wonder if anybody in the audience could actually translate this for me. I happened to be at a John Vane meeting, and I'd handed in my slides to the projectionist. And I started giving my talk at the Royal Society, and virtually all my slides were converted from Arial font to Wingdings. So I had to give an entire presentation trying to guess what my slides were labeled with. Anyway, this is what it's supposed to look like. But it's well known that prostacyclin synthase is bimodally distributed in cells, and that it has a perinuclear expression. And prostacyclin is known to bind to these transcription factors called peroxisome proliferator activator receptors. And they're also known to be activated by a variety of different agents, including metabolites giving rise to lipoxygenase metabolites, as well as prostaglandins like prostaglandin J2. What is the role of these transcription factors? Certainly, well, we know that they regulate all isoforms of PPARs. There are 3 of them, alpha, beta, and gamma, cause vasodilation. They regulate cell proliferation and also regulate activation of adhesion molecules. It was also known in the late 90s that some prostacyclin analogues could also bind to PPARs. And we were instrumental in also showing that the IP could phosphorylate these PPARs, although we're not sure the mechanism of this activation. So what about the role of the IP receptor in normal pulmonary arteries and also diseased arteries? Well, in normal arteries, we can, this is treprostinil, and these are small rat pulmonary arteries. The treprostinil relaxation is almost entirely dependent on the IP receptor. Not shown here is if you give a PPAR beta antagonist, you can also inhibit this relaxation. So we think there's IP receptor activation of the PPARs. In veins, the picture is slightly different, so that you use the same prostacyclin receptor inhibitor, and it only partially inhibits the relaxation. And we think this is activation of another prostanoid receptor. Okay, so if we look at the binding affinity of the different agents that are used to treat pulmonary arterial hypertension, prostacyclin, iloprost are the most potent, and beroprost and treprostinil are about 10-fold less potent, and then the prodrug, the selective egg metabolite is somewhere in between. However, this is work done by Xavier Nerel in Paris, but if you compare the relaxation properties of these different analogues, this is what you get. So iloprost is the most potent relaxant of large pulmonary arteries, and then treprostinil, as you predict, is about 10-fold less sensitive in terms of causing relaxation. But what's curious is that the selective egg metabolite is about 70 or 80% fold less active. And this was kind of a surprise, really, given the sensitivity of the IP receptor. However, we showed that the selective egg metabolite looks like it's behaving like a partial agonist. So here is the cyclic MP properties of iloprost, and this is MRE. So not only is it less potent, but it's also less efficacious. Now, what that means clinically is really not known. So if you look at normal human pulmonary arterial smooth muscle cells, and then you look at cell proliferation induced by serum, and this is the anti-proliferative effect of treprostinil, as well as iloprost, it's almost completely inhibited in the presence of the IP receptor antagonist. So at least in normal cells, it's behaving how you would predict. And this is Emilia Forsetti, who was a PhD student in my lab, who did a lot of the initial work. So what about cells from patients with pulmonary arterial hypertension? Well, just the first thing to remind people is that if you isolate the cells from patients and compare it to normals, the cells from children or adults with pulmonary hypertension, they grow, here's the adult and here's the child, they grow much more aggressively than cells grown in the same conditions but from either an adult or a child. And this suggests that the phenotype of these smooth muscle cells has been completely altered. And these are growth rates that would exceed a lot of cancer growth rates. So if you look at the selective egg metabolite, here is this is the dose-dependent anti-proliferative effect of MRE, 269, sorry, it's actually shown here as its other name. and its antiproliferative effect is completely inhibited by the IP receptor antagonist. So clearly the IP receptor is working in the patient cells. But when we looked at the effect of treprostinil, another analog given to treat patients, the IP receptor antagonist really doesn't do much. And so we were sort of left scratching our heads a bit until we made this discovery. That if you compare the binding affinities of iloprost with treprostinil, the profile is completely different. If you look at the EP2 receptor, here is treprostinil. It has a Ki at the EP2 receptor of 3.4 nanomolar. So actually, it's tenfold more active at the EP2 receptor than at the IP receptor. Now, those of you who are pharmacologists will know that the hill slope of this binding curve is 1. So that's in my title, slopes. But if you compare the effect of iloprost at the EP2 receptor, it really is quite less active at this receptor. And then we looked at another receptor, the DP1, and treprostinil was a potent activator of the DP1 receptor, the potent binder. So what about the expression of prostanoid receptors in pulmonary arterial hypertension? But first of all, we wanted to look at what the expression profile was in normal cells. What you can see is that the IP receptor and the EP2 receptor are expressed what we think is sort of fairly sort of unity. But if you looked at the message levels in the cells in PH patients, we have a completely different profile. We lose expression of the IP receptor, and we gain markedly, we get an upregulation of the EP2 receptor in pulmonary arterial hypertension cells. There's a precedent for this kind of behavior of prostanoid receptor activity in that there's a paper published showing enhanced EP2 receptors in asthmatic patients. So what about EP2 receptors? Are they functional in PH patients? Here is the EP2 agonist, butaprost. And you can see that in cells in PH patients, it is potently inhibiting cell proliferation. What about treprostinil? Well, we used a pretty selective EP2 antagonist made from Pfizer, and we get this huge rightward shift in the dose-response curve to treprostinil, suggesting that EP2 receptors are driving the antiproliferative effects of treprostinil. And if you give an IP receptor antagonist on top of that, we can get a further shift. So we think that the majority of the effects in terms of proliferation are EP2 receptors being activated by treprostinil with some contribution from the IP receptor. So we decided to knock down the EP2 receptor using small interfering RNAs. And what we find is that the antiproliferative agent effects of treprostinil, both at 10 nanomolar and 1 micromolar, are almost completely inhibited by knocking down the EP2 receptor. And then we also wanted to look at histological slides of small pulmonary arteries, both from normal patients and patients with pulmonary arterial hypertension. What you can see is that the expression of the EP2 receptors is almost confined to the endothelial cells in normal tissues. But if you look at pH, blood vessels, there's a strong expression of the EP2 receptor in the smooth muscle and endothelial cells. And the grey images are really converting the brown staining into a grey image. And this is really the analysis of staining of the EP2 receptor, and it's correlated per area of staining. And what you can see is the EP2 staining goes up, as well as alpha smooth muscle actin. That's what you'd expect when a blood vessel gets thicker. But if you were to look at the ratio between EP2 to smooth muscle alpha actin, there's more that goes up, and there's more EP2 receptor expression per unit area of muscle. So I've got a couple more slides, just looking at the role of the PPARs in the anti-proliferative effects of prostacyclin analogues, just to try and bring you back to the story about PPARs. So what we found, looking at the expression of PPAR gamma in small and in larger and smaller arteries, is that the expression in the medial layer goes up in pulmonary arterial hypertension. And we were able to reverse the anti-proliferative effects of treprostinil with a reasonably specific PPAR gamma antagonist. So what we now need to make the link is whether the EP2 receptor is activating our PPAR gamma. And then finally, to bring back the calcineurin story. This is calcineurin A beta, the active subunit of calcineurin. The catalytic subunit goes up in response to serum. And then if you pre-treat your cells with treprostinil, both the expression and the nuclear activity of calcineurin A beta goes down. And we can reverse this effect with a PPAR gamma antagonist. So we think that calcineurin is suppressing PPAR gamma function, but that treprostinil can reverse these effects. And this is just the mean data showing nuclear translocation, which means activation of calcineurin A beta, going up with growth, going down with treprostinil, being activated by a PPAR gamma ligand, and then being reversed by the antagonist. I've come to pretty much the end of what I wanted to tell you. So this is me on a slope. I absolutely adore skiing in deep snow. This is me skiing. I just wanted to leave you. I just wanted to dedicate this lecture to Cornelia Clapp. I think that her character personifies, everything about being a teacher, a mentor, and a great academic. What Edwin Cochrane said about her, he was a famous embryologist. He said that she was a great influencer of character of individuals, and that that her effects, are seen from generation to generation. Thank you very much.